Dr. David Potter realizó sus estudios de licenciatura en biología en MIT. Recibió su título de médico y doctorado de la Universidad Johns Hopkins. Se formó en medicina interna en el Centro Médico de la Universidad de Stanford y en hematología y oncología en el Centro Médico Tufts, New England. Finalmente realizó estudios postdoctorales en el Centro para la Investigación del Cáncer del MIT en el laboratorio del Dr. Philip Sharp. Ha recibido diversas distinciones tanto por su investigación como por su participación en la formación de estudiantes, entre los que destacan el Premio Walter por su investigación en cáncer y distinción en enseñanza por la Universidad de Tufts. Recibamos con una grata bienvenida al Dr. Potter.
Thank you very much for your very kind hospitality. I want to thank Dr. Luis Herrera and Alfredo Cantu and all the organizers of this meeting for the opportunity to present some of the work that we have ongoing in our lab right now. And I also want to thank the students who have very generously been sent by INCAN to work on this project. And I want to first start out with an introduction about the molecule metformin. And so, as you know, metformin is the most widely prescribed diabetes drug in the world. And there's tremendous interest right now on whether metformin can be repurposed for either cancer treatment or for cancer prevention by interfering with the function of bioenergetics and possibly cancer cell mitochondria. There's interest in whether it could be an exercise mimetic, and there's interest in whether in breast cancer patients it could be used to prevent recurrence of breast cancer. And so there's a clinical trial that's ongoing right now, sponsored by the National Cancer Institute of Canada and the National Cancer Institute in the United States, to see if metformin is given to patients who have had primary treatment with breast cancer, whether the metformin will reduce the rate of recurrence. And so we're waiting for that trial to report in the next 4 to 5 years. So this slide summarizes what I'm going to tell you about today. And we've been working on an enzyme which metabolizes about half of prescription drugs in patients. That's cytochrome P450-3A4. And this enzyme we have found unexpectedly to be involved in breast cancer growth. And I also want to draw a connection with work that's being done actually here in Mexico on cytochrome. Okay, thank you. And there's work that's ongoing. It was published by Dr. Florio Sanchez in 2014, showing a connection between CYP3A4 and risk factors for breast cancer. And so this is an area of active investigation. So on the left side, what you can see is a cytochrome P450 nanodisc. And this is a contraption that kind of looks like an hors d'oeuvre, a cracker with an olive sitting on it. The olive is the enzyme, and the enzyme is able to metabolize whatever you feed this, this contraption. And it allows us to definitively determine whether a substrate is processed by the enzyme. It also allows us to tell whether an inhibitor, a prescription drug, for example, may interact with that enzyme and prevent its function. This work then led to the centerpiece, which you can see there, which is a co-crystal from a group at UC Irvine, Irina Sevrakova and Tom Poulos, who were able to crystallize metformin in the active site of CYP3A4. And then what this led us to do, working with a Minnesota supercomputer and our College of Pharmacy, Elizabeth Ambrose and Gunda Georg were able to help us find and design and synthesize a better metformin, hexylbenzyl biguanide, and you can see that molecular structure there on the right. So I'm going to tell you today about the journey from the beginning to the development of this new CYP3A4 inhibitor. So this slide summarizes what we think the mechanism is, and what I'll do is I'll tell you in detail how we got there. But basically what we're thinking is that this cytochrome P450 enzyme, which is normally found in the liver, is somehow co-opted by cancer cell mitochondria. And unexpectedly, it's sitting on the mitochondria and orchestrating the function of the electron transport chain. And what we think is happening is arachidonic acid on the left is being metabolized to an epoxide on the right. And then this epoxide is now interacting with the electron transport chain and facilitating its function in cancer cells, perhaps giving those cancer cells some metabolic advantage and ultimately driving mTOR and biomass assembly in the cancer. And so the main points of the talk today are, number one, that CYP3A4 is an arachidonic acid epoxygenase required for breast tumor formation, and I'll show you data on that from genetics. CYP3A4 suppresses autophagy, or the breakdown of biomass in breast cancer, in part by inducing AMPK activation, and AMPK is a major arbiter of bioenergetics. And then CYP3A4 epoxygenase activity promotes the mitochondrial electron transport chain, and I'll show you some of the data supporting that. And finally, I want to say that metformin inhibits breast cancer unexpectedly, in part by inhibiting this process and the cytochrome P450 epoxygenase activity. So the questions that we're going to address today are, number one, does cancer cell intrinsic CYP3A4 epoxygenase activity regulate breast cancer growth? So what we're really talking about here is exogenous versus endogenous. So a lot of interest in the EET or epoxygenase mechanisms have really dealt with the idea in the blood vessels. And Judah Folkman's group at Harvard Medical School and Deepak Panigrahi, one of his students, has been a major force behind this model. So what we're adding to this model is that not only are the CYP enzymes important in the formation of new blood vessels in breast cancer, but also that these enzymes are expressed within the cancer cells themselves and promote the growth of the cancer cells. And secondly, what we want to know is whether this epoxygenase activity is affecting mitochondrial function. Is it affecting oxygen consumption rates or lactate production or both? And then does metformin inhibit this enzymatic activity? And then finally, whether structural biology could offer an avenue to the design of a better inhibitor of CYP3A4 epoxygenase activity. In other words, can we find a better metformin that's tuned to the specific purpose of treating cancer? So this is a beautiful diagram from Deepak, and it shows the work that his lab did on CYP2C and CYP2J, which are at the bottom of this slide. And then the work that we did on the CYP2C, CYP2J, and CYP3A actually within the cancer cells themselves. And so what are these EET molecules interacting with? We don't really know. There's a lot of controversy right now because a true EET receptor has not yet been characterized. But what we do know from work at Vanderbilt, from Ray Harris and colleagues, these EETs are interacting somehow with renal tubular epithelial cells, stabilizing them and protecting them from toxic insult. And we know from John Subert's work at University of Alberta that these EETs are involved in the health of the vascular system, in myocardial muscle and vascular smooth muscle cells. And we also know from the work of Jorge Capdevila at Vanderbilt that these EETs are playing an important role in the regulation of blood pressure. However, with EETs, all politics are local. The EETs are made locally and they seem to be having activity locally, meaning that they seem to be made in the body where they are needed, and not so much as endocrine factors, but more as autocrine and factors. And these EETs have been screened for interaction with G protein-coupled receptors. Bill Campbell's group at University of Medical College of Wisconsin has been working on this, and also Alkiad's group has discovered that there are G protein-coupled receptors of the prostaglandin receptor family listed on this slide, which, when expressed in Xenopus oocytes, in frog oocytes, basically are sensitive to EETs, and they can identify individual receptors. However, these receptors all have low affinity for EETs, suggesting that much of the activity of the EETs may be very local. And so this is where Mexico comes into this story. So Dr. Floriana Sanchez, working at the Women's and Babies Hospital here in Mexico City, was able to find a relationship between CYP3A4 expression in breast cancer and its association with risk factors in Mexican women. And at this very same time, we were collaborating with Dr. Young Bae at Daegu, Korea, looking in a tissue microarray from Korean patients, and essentially we found very similar results. So these are converging science coming from different patient populations, different parts of the world. So this is the results that we got from the Korean tissue microarray. And what we found is that CYP3A4 is not only widespread in breast cancer, but its correlation is with estrogen receptor alpha. So in collaboration with a group at University of Michigan, we used aqua immunofluorescence to compare nuclear estrogen receptor alpha with cytoplasmic and CYP3A4. And what we found is a very strong correlation between these. And this suggested to us that a lot of the biological work that we do in vitro and then in animal models should be focused on estrogen receptor positive breast cancer. So Dr. Zijun Guo in our lab developed a beautiful mass spectroscopy assay for EETs. And he was able to show, without any question, that CYP3A4 could make EETs. And he settled a controversy that had been going back about 15 years. But with more sensitive mass spectrometry methods, we were able to demonstrate that indeed these EETs are made. And not only that, with Dr. Ian Blair at University of Pennsylvania, we were able to show that the synthesis was chiral, meaning that there was a specific enzymatic bias and that this wasn't nonspecific oxidation. And we also found that if you knock down the CYP3A4 gene and protein in breast cancer cells, you can lower the amount of EETs in those cancer cells and lower their proliferation. The other thing that Dr. Guo did, which was a beautiful piece of enzymology, is that he measured the oxygen Km, Michaelis-Menten constant for oxygen, and it turned out it was right in the range where there would be motive and opportunity for the CYP3A4 to participate in cancer growth, meaning that it was right in the range where tissue oxygen may be in cancer. So we then contacted one of our friends at the University of Illinois, Urbana-Champaign, Steve Sliger and Ilya Denisov, and we asked whether they could use these CYP3A4 nanodiscs to measure the spectral association between drugs and these nanodiscs, because this could give us an idea of which drugs out there might be interacting and could potentially inhibit the production of these EETs made ectopically in the cancer and might be promoting the growth of the cancer. And so the nanodiscs, like the microsomes, made EETs in an NADPH-dependent fashion. And what Zijun was able to show in a JBC paper in 2011 is that when you did the knockdown and you added back the EETs, you could restore the growth of the breast cancer cells. And not only that, but STAT3 was a very important part of this story, and that if you knock down the CYP3A4, you lower phospho-STAT3 in the nucleus, and then when you give back EETs, you restore the phospho-STAT3 in the nucleus. So we then wanted to know, does CYP3A4 affect breast tumor growth? And so this experiment takes the knockdown cell lines and we introduce them into the mammary fat pad and asked in a nude mouse model whether MCF7 estrogen receptor positive, HER2 negative breast cancer cells would grow if we knock down CYP3A4. And the answer is they don't form tumors. And the black line that you see along the bottom of the panel on the left shows the failure of those tumor cells to form tumors. The control tumors initiate tumor growth without any difficulty, and that's shown with the white triangles. And if you look at the tumors, you find that the tumors are growing with CYP3A4. However, there are still tumors formed in the knockdown mice, but however, these tumors don't form large structures. They don't grow beyond several hundred microns in size. And so what's happening here is that the knockdown of CYP3A4 isn't preventing the engraftment of the tumor cells, but it's preventing their growth to form tumors. So we then wanted to know, if that's the case, maybe there was a problem with biomass accumulation. And we paid attention to some work that John Subert's group was doing on mitochondrial biogenesis. And this made us start to think that these EETs, which may play a role in cardiac mitochondrial biogenesis, might also play a role in the building of biomass and protection of cancer cells. And furthermore, it was thought that these EETs may somehow induce autophagy in cardiac myocytes, and this generated some confusion in the field. But then we realized that the AMPK alpha subtype is different in cardiac tissues compared to breast cancer. And so the behavior of the AMPK is quite different. So we've then asked, is the CYP3A4 present in breast cancer mitochondria? And what Zijun found is that indeed CYP3A4 is present in the breast cancer mitochondria, and you can see mitotracker staining mitochondria in red. The mitochondria are in the perinuclear area, and then a polyclonal antibody staining with a FITC secondary antibody. You can see the CYP3A4 location, and when you overlay the two, they match in terms of their location. And when you overexpress CYP3A4, you find the same result. So if you do tenfold overexpression of CYP3A4, you see more correlation with the CYP3A4 and the mitochondria. And not only that, but these breast cancer cells grow more quickly. So then we went to the seahorse machine to ask what's happening to oxygen consumption rates and to extracellular acidification. And so this machine is able to measure oxygen consumption in real time, and it can simultaneously measure the production of acid, which is lactate, in the medium of the cells. So you can then add various chemical probes and ask the question, what do these chemical probes do to oxygen consumption and lactate production? One of the problems working with EETs is that they're easily hijacked on the way to the target. So if the EETs are put on cells, you can see biological effects at the plasma membrane and plasma membrane signaling. But what you can't tell is what's going on in the mitochondria. And this requires two strategies. One thing that we can do is use what's called an epoxide hydrolase inhibitor, which is foreign to the cell and helps stabilize the EETs throughout the cell. And when we do that, what we see is that oxygen consumption rates increase, but there's not much difference in terms of the extracellular acidification. So this told us immediately that we should be focusing primarily on how these EETs are affecting breast cancer mitochondria. And then, in collaboration with Camille Falk at UT Southwestern and John Immig at Medical College of Wisconsin, they very kindly shared an EET analog which was known to have the same cardiovascular effects as EETs. And we put this on the breast cancer cells, and what we found was really quite astonishing, because we were actually able to increase the oxygen consumption rate and metabolic rate of these cells. On the other hand, we also noted that there was a reduction of the extracellular acidification, meaning that these cancer cells had now shifted more away from glycolysis and more towards aerobic respiration. So we figured that we had some idea of a handle or a switch that could potentially regulate mitochondrial function. And when we added a EET antagonist, what we could do is suppress the oxygen consumption rates and also suppress the extracellular acidification. So then the question came, what does the CYP3A4 affect AMPK alpha, which is a major energy broker in the cell, albeit an indirect energy broker? And what we found is that when you knock down CYP3A4 stably, AMPK comes up, the activity goes up. When you take the product of the CYP3A4 and put it on the cancer cells, the phosphorylation of AMPK goes down. So this told us that the CYP3A4 was somehow able to pull strings and levers that could affect biomass formation. And in fact, when we knocked down CYP3A4, what we found is that these cancer cells were burning the furniture. they were in a general catabolic state, which meant that they were unable to build biomass. And I think that one of the emerging themes, whether you're interested in mTOR inhibitors or other aspects of bioenergetics to block cancer cell growth, the underlying theme is that if you can't build biomass, you can't build a cancer. And so what has happened here is that the cancer cell is basically trying to save itself. And so it's turned on the aerobic respiration, it's turned on glycolysis, and it's doing everything that it can to increase energy stores. But money isn't everything, because the cancer cells have a plethora of ATP, but they can't grow because they've lost the control of information and signaling that's necessary in order to build a biomass. And the AMPK activation artificially with ACAR does a similar thing. And so it activates OCAR, it activates ECAR, it says start respiring, burn the furniture, but the cancer cell is unable to survive. And in a very nice experiment, Fernando Luna and Adela Galvan worked on basically showed that these cells that are overexpressed, that have knockdown of CYP3A4 are actively in autophagy. And the P62 regulator of autophagy that gets consumed with autophagy is reduced, and the ratio of LC3B2 to B1 is increased, consistent with an autophagy state. So the question then is, can metformin substitute for the CYP3A4 knockdown? So what did we know about metformin? We knew that metformin activates AMPK. But then the question was, does metformin inhibit CYP3A4? And when I called the Poulos lab, they basically said, no, it's not going to fit in CYP3A4 because it doesn't have the right charge. It's extremely unlikely that that's going to happen, but we'll try it anyway. What the heck? And so, in fact, what happened is that metformin did inhibit the CYP3A4 function. So we also got a clue that this may be the case because there was a group in Korea that had shown that metformin interfered with nifedipine metabolism in the liver. And Ann Thor's group at the University of Colorado had shown that metformin, just like the CYP3A4 knockdown, inhibited STAT3 in breast cancer. So when Zijun tested this, he indeed found that metformin inhibited EET biosynthesis in microsomes and in cells. And so just like the knockdown, he was able to lower the amounts of EETs made, and he was also able to, with microsomes in vitro and also in cells. However, the amount of the concentration he had to use is industrial strength. He had to use 5 millimolar metformin in order to block this. But this is just like the experiments published by others, such as Michael Pollack's group at McGill, where they were able to inhibit cancer cell growth and block mTOR pathways, but they required millimolar concentrations of metformin. And so we sent metformin to Steve Sliger's group, and we were able to get a Surrey spin shift, and we could find a dissociation constant with CYP3A4 of about 400 micromolar. And then Tom Poulos sent an email to me saying, Christmas present, we found a docking. And so he sent this structure to us, which was very unexpected. So metformin is docked in the active site of CYP3A4 right over the heme ring. It interacts with arginine 212, and it's right in the position where it should interfere with metabolic functions of CYP3A4, as was predicted by the interference with nifedipine metabolism. However, one of the interesting things is we also tried ritonavir, which is an excellent CYP3A4 inhibitor, but the arginine 212 was 19 angstroms further away from the heme iron, and the ritonavir was in a different location. So what this told us is that it was impossible to predict a priori of which inhibitor we should be using. But the beautiful thing about metformin is that it is a click chemistry module in that you can put different moieties on the left side and the right side of metformin and make an entire myriad of different molecules that can be tested for activity. So we were very excited when we saw this. So what we wanted to do then was go to an array of compounds that were in the patent literature, because we weren't in the business of running a pharmaceutical company, and what we wanted to know was what had other people done, and could we do in silico docking with the Minnesota supercomputer to see which of these compounds would have a good dock score. And the good thing about these compounds is that they were all in a feasible chemical space. Somebody had already made them, and they'd done NMR structure analysis, so the ground was open to start hunting. And so in this hunt, we identified the molecule in the bottom left-hand corner, hexyl benzyl biguanide, which was 2.5 orders of magnitude better as an in silico docking candidate compared with metformin over there in green. So the dock constant was for metformin 4.75, for HBB 7.11. There were a couple of other molecules. We made some of them to test as well, but as it turned out, nothing was better than hexyl benzyl biguanide. And when we looked at the binding to these nanodiscs, now we had lowered the spectral dissociation constant fourfold, and now we knew we were in the right ballpark. So then what Sijun did was he took the HBB molecule and he took microsomes and he asked, what is the IC50 for the biosynthesis of EETs? And what he found was very encouraging. So for CYP3A4, now it's 9 micromolar. With metformin, it was 5 millimolar. So we're really moving in the right direction. And also what we wanted to know is there specificity? So he took a look at the vascular CYP2C8, 50 micromolar. So it's higher. And then he looked at CYP2J2, also vascular. There was no inhibition. And then he wanted to look at CYP4A11, again, minimal inhibition in the hundreds of micromolar. So now we were in a lucky position because we had a molecule with selectivity. And this is not a trivial point, because if you look at other potential targets where we'd like selectivity, there's a tremendous interest now in immunology of breast cancer. CYP3A4 isn't expressed in lymphocytes. And the 2J2, which is expressed in lymphocytes, isn't sensitive to HBB. So now if we want to interface this kind of approach with immunotherapy methods, we have some ideas that we could have potentially some selectivity. So this is what happens when you take a range of breast cancer cells and you expose them to a range of biguanides. So if you look ER positive, HER2 negative, MCF7 cells, and you look at metformin, buformin, fenformin, it's milli, milli, milli. Then you look at HBB, and you're in the micro range. If you add back the ETs, the upper curve is partial restoration of growth, by adding back exogenous EETs. We know this isn't going to be perfect because we can't restore the EETs that are in the mitochondria. So the foreign molecule can get into the mitochondria, but the natural molecule can't get in so well. When we add the unnatural C22 analog, we do a little bit better at restoring growth. Now, what was also interesting from this whole ER-positive question, which came from Floriano Sanchez's work and with the collaboration with Bei Young, what we find here is that in the triple-negative breast cancer, we don't see activity in terms, we see activity, but we don't see rescue with the ETs. So we think there's something going on here, but it's not rescued. However, when we add N-acetylcysteine, we see the opposite. So N-acetylcysteine can rescue the triple-negative breast cancers partially rescue them from HBB, but the N-acetylcysteine has no restorative effect on the ER-positive MCF7. So now this told us that we had a biology and we had some ideas about the currency that's involved in this biology. So the next thing we wanted to do was to see what happens to ATP levels and metabolism when we bring in HBB instead of metformin. So this is a control experiment with metformin. We see minimal effects on oxygen consumption rates, minimal effects on the extracellular acidification, and no effect on ATP levels. Then when we add HBB at micromolar instead of millimolar concentrations, what we're seeing is a profound effect on oxygen consumption rates and on extracellular acidification, and ATP levels are diminished. And we see the same effect in other ER-positive and triple-negative breast cancer cell lines. And when we look in detail of what the HBB is doing, it's doing everything that we would expect an inhibitor of CYP3A4 to do. It's inhibiting mTOR, albeit later. It's activating AMPK, albeit later. But it does have the immediate early effects on STAT3, which we would expect. And these effects occur either in the presence or absence of serum. And so then we went to an animal model. And here we had some challenges because it turned out we had invented a weight loss drug. And the mice really didn't tolerate this drug very well. And so instead of using metformin at between 50 and 200 milligrams per kilogram per day, we were looking at 2, 4, 6, 8 milligrams per kilogram dosing. And we found a minimal effective dose with daily dosing, which was about 4 milligrams per kilogram. And so effectively, this dosing is more than 100 times lower than what you would where you'd give metformin. But we don't see any effect on triple-negative breast cancer in the animal model. Now, surprisingly, we activate AMPK, but there's absolutely no effect on tumor growth. And what this is telling us is that AMPK is secondary and that we need to get closer to the target to understand how to inhibit breast cancer energetics. So then what we did was we shifted the dose and schedule, and we were able to give a total of 24 milligrams per kilogram per week in 4 divided doses, and we were able to get some inhibition of growth of the breast cancer. Now, if you're on the editorial board of clinical cancer research or cancer research, you would look at this and you say the glass is half empty, because how are you going to translate this to the clinic? However, I would argue that you cannot make a conclusion a priori from monotherapy in a mouse model and how to develop a drug in the clinic. And there are drugs that have made it into the clinic that have been successful, that have had modest activity in the mouse model. So I think that the jury is still out. But what we did find is that the tumors were half the size compared to control, and we were able to manage the weight loss question. So the question then is, moving forward, how to develop HBB. And I think that the way we need to develop it in the clinic initially is as monotherapy, because if we have to be able to walk before we can run. It may very well be that you could combine HBB with all kinds of things and get good results, but you have to know how to handle the drug as a monotherapy. You need to know what the toxicities are, what the ADME is for the drug, how it's metabolized, and before you can really move forward. And so this leaves us with a model for how the cytochrome P450-3A4 is playing a role in breast cancer. And we can see from in the top, there are two sites of action. One at the plasma membrane, where the EETs are driving STAT3 and mTOR activation. And this is an immediate early effect. I haven't shown you all of the data from that. Some of it is previously published. But now, when you look at the mitochondria, we have a second site of action. And so this CYP is sitting on the mitochondria, and it's overlaying the regulation of the electron transport chain. When the CYP is functioning well, it's increasing oxygen consumption rates, it's increasing ATP levels, and it's suppressing AMPK activity. And so in this situation, mTOR is unleashed and able to build biomass. When you look at the case of HBB, at the membrane, the HBB can block EET biosynthesis. It can block the immediate early activation of STAT3, and it can block late signaling to mTOR. At the mitochondria, what it can do is lower oxygen consumption rates, decrease ATP levels, activate AMPK, and inhibit mTOR function. So the way forward in the laboratory is to move forward to immunocompetent mouse models. And what we want to know is whether the CYP sensitivity in breast cancer is only in the cell lines we're studying in culture, or is it also true in animal models, and is it true in mouse CYPs that might be playing a role in mammary carcinoma growth. And so what we've done is we've done a CRISPR-Cas9 gene excision of CYP3A11. The cells grow more poorly, and unfortunately the antibodies for mouse CYPs are terrible, and so we're in the process of making our own antibodies. But what we have now is a clue that these CYPs are found in mitochondrial fractions, and the CYPs of great interest are 3A11 and 2C44. And so how general are the effects of HBB? Will they inhibit mouse CYPs? And in fact, HBB does inhibit mouse CYPs. And so if we compare metformin and HBB in an E0771 mammary carcinoma model in C57 black 6, what we're able to do is the same thing that we could do with human mammary carcinoma cells. So this is saying to us that there's enough homology between the mouse CYPs that are promoting mouse tumor growth that we can block with the same processes with HBB. And it turns out that the mouse mammary carcinoma cell lines are very much inhibited by HBB, and the IC50s range between 2 and 30 micromolar, and so there are a number of different animal models that we can work with that are both ER positive and basal in type. And furthermore, HBB suppresses the EETs in the ER positive mouse mammary cancer cells. So what this is saying is that these findings really cross species boundaries, and what they allow us to do then is to do genetics in mouse models. So in summary, I hope I've shown you today that CYP3A4 is an arachidonic acid epoxygenase required for breast tumor formation, that CYP3A4 suppresses autophagy in breast cancer, in part by inhibiting AMPK activation, and thus could contribute to biomass assembly, that CYP3A4 arachidonic acid epoxygenase activity promotes the electron transport chain, and that metformin inhibits breast cancer in part by inhibiting the structural target CYP3A4 and its cognate arachidonic acid epoxygenase activity. So I want to thank INCAN and the wonderful students who have come to our clinic. You know them all. And also to our laboratory, and this has been a wonderful learning experience for me as well as for the students. And I want to congratulate the students on the cell chemical biology paper on this subject, which is coming out October 19th edition, and you guys got the cover of Cell Chemical Biology. And I want to thank again, INCAN. I want to thank Luis for his very kind support, and Alfredo for his very kind support. I want to thank Erwin, Sebastian, Victor, Cesar, Fernando, Diego, Mariel, and Adela for all of your great work in the laboratory. And I went past, and I want to also thank the University of Minnesota, the NIH, Susan G. Komen Foundation grants, and other flight attendants Medical Research Institute that got us started on this work, and our Hematology, Oncology, and Transplantation Division, and also the Randy Shaver Cancer Research and Community Fund, which has given very much support to our work. So thank you for the opportunity to speak.